All right, here we go. Awesome, awesome, amazing, wonderful AP Bio 2 kids. This is our last biomolecule lecture. I know you're so excited to wrap up these biomolecules. So the last one we're going to talk about is nucleic acids. Okay, so we've already finished carbs, lipids, and proteins. And if you remember correctly, as far as the elements that each of those contain, carbohydrates contain C, H, and O. Lipids contain C, H, and O, but as far as the ratio, the carb is a 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. Okay, 1 to 2 to 1. Had a little mistake there. Fix that. 1 to 2 to 1. The lipids do not have a 1 to 2 to 1 ratio. Remember, it's much greater than that. And then if it's a phospholipid, we are going to see a phosphate group if it's a phospholipid. Other than that, though, you're not going to see that. Proteins, don't forget, we have C, H, O, N, and sometimes sulfur, especially if it's cysteine with that sulfhydryl functional group. So now on the last one, nucleic acids, we are going to see carbon, hydrogen, oxygen, nitrogen, phosphorus. So look at the group that we now have present with our nucleic acids. Okay, this one is going to fit into the perfect little monomer building to form polymers. So we're going to see the same kind of pattern that we saw with carbohydrates. So just really quickly, if we could visit that, remember a carbohydrate, the monomer is a monosaccharide. Okay, the polymer is going to be our polysaccharides. Okay, if it's a protein, remember the monomer is an amino acid. The polymers are going to be polypeptides. Okay, go back again. In the carbohydrate, if there are 2 of them, those are disaccharides. In the protein, if there are 2 of them, they are dipeptides. The lipid group, remember, was unique because they are very diverse. They do not have monomers and polymers. So now on our last group, we're back to this monomer building a polymer. Okay, so the monomer, I'm going to skip and go down to this bullet right here. The monomer is called a nucleotide. It sounds nice because it kind of sounds like nucleic acid. So this is our last and final monomer. It's called a nucleotide. And those build up to show our examples of our polymers. Okay, so the first polymer we have is DNA. You should know that's deoxyribonucleic acid. That's our main information molecule. It's your genetic information. It's everything about you. Okay, there's also a polymer known as RNA. RNA is the one that helps out DNA in making the proteins that we need for all the reactions in our bodies. Okay, the last one is actually not a polymer. ATP is not a polymer. It is a monomer, which is kind of interesting. It's a monomer. We'll talk a lot about ATP when we get to cellular respiration and photosynthesis, when we get to those units. But ATP is a nucleotide. The weird thing about ATP is it's an energy molecule. That's the one that is energy. So if I ask you what are functions of nucleic acids, surely you would be able to tell me genetic information and making proteins because of DNA and RNA, but then also let's not forget our ATP, which is a single monomer, but it's used for energy purposes. That's like our cell energy that we make through cell respiration. Okay? All right, now we're going to examine a monomer more closely. I think I'm going to change colors. I'm tired of this green. We're going to examine a monomer more closely. So if you see what a monomer is made up of, okay, you can see a perfect picture right here. Here's the monomer, which is a nucleotide. Okay, so here's your nucleotide. The 3 parts are, we have a pentose sugar. Remember, that means 5-carbon sugar. You can see it right here. It's shaped like a pentagon. Okay, it's either going to be deoxyribose if that sugar is in DNA, or it's going to be ribose if that sugar is in RNA. Okay, if it's ATP, it's going to be ribose sugar. ATP is a lot like RNA. Okay, so part 1 is a 5-carbon sugar, either called deoxyribose or ribose. Part 2 of a nucleotide is a phosphate group, and part 3 is a nitrogenous base. And we'll talk a little bit more about that in a minute. But I just wanted to show you, if you go back up here and look at your letters that are found in a nucleic acid, C-H-O-N-P, if you examine this monomer right here, you can see where those letters are derived from. The sugar, the pentose sugar, is where the carbon, hydrogen, and oxygen are found. The phosphate group is where the P comes from. The nitrogen base is the N. So I have a sugar, okay, which is technically, this is a carbohydrate, and don't let that confuse you. I know we're talking about nucleic acids, but it's a very big group, and its monomer has a carbohydrate as one of its components. Another component is a phosphate, and a third component is a nitrogen base. Okay, now, if I want to take one of these monomers, you can see it right here, because here's the nitrogen base, there's the sugar, there's the phosphate. If I want to add that monomer to a second monomer, I'm still going to use dehydration synthesis. So what am I going to lose, guys? That's right, I'm going to lose a molecule of water, and I'm still going to get some covalent bonds. Okay, so now we have a different name. Oh my goodness, here we go. Remember the name of the bond. If it was a carbohydrate, the name of the bond between the monosaccharides was a what? Glycosidic linkage. If it was between a lipid, the name of that bond was called an ester linkage. If the dehydration synthesis happened and we got we had a covalent bond between two amino acids in a protein, it was called a peptide bond. Now our last bond for this group has a really weird name. It's called phospho. Okay, here's a P-H-O, phosphodiester, D-I-E-S-T-E-R. So it's a phosphodiester linkage, which is between two nucleotide monomers. Okay, now if I get all this stuff off the screen, I can show you. That looks much better, right? It's kind of clean. I can show you where that is on this picture, okay? So here is one. I'm going to circle it. There's one monomer. Okay, that is my monomer. So that's my nucleotide. Here's my second one right here. So between those two, if I change colors to red, here's the bond between the two right there. That bond is my phosphodiester linkage. I'm going to abbreviate it. PL. That's my phosphodiester linkage. There's another one right here. There's my phosphodiester linkage. There's another one right here, and another one right here that would be between the next monomer. So once again, a phosphodiester linkage is a covalent bond between nucleotide monomers that's part of the nucleic acid biomolecule group. Okay? My monomer is a nucleotide. You can see it right here. The three parts are 5-carbon sugar, nitrogenous base, and a phosphate group. So that makes my elements C-H-O-N-P. That's where I get the element. Okay? Now, all we have to left to do, this is going to be really short, is just kind of look at some of the basics of parts of the nucleotide. So this slide, guys, look at the name of it. This is a nitrogenous base slide. So this is saying in my nucleotide, okay, in my nucleotide, one of the components is a nitrogenous base. So there are some choices. This is kind of an elaboration of my nitrogenous bases. So here they are. Okay, there's 1, 2, 3, 4, 5 of these, because it depends on if it's DNA or RNA. I'm sure you remember a little bit about our rules. Do you guys remember when we did DNA and RNA in freshman year, and we did A to T, G to C, but then we had, if it was RNA, there was no T, that was replaced by uracil. So those are our 5 nucleotides. We have an adenine nucleotide, guanine, uracil, thymine, and cytosine. But if you look at their shape, these 3 down here at the bottom all have a single ring. Okay, it's a single ring structure. These 2 up here have double rings, like that's the adenine, here's a double ring, and that's the guanine. So their shape is a little bit different, so what we do is we put them into 2 categories. We call these are the purines. These down here are the pyrimidines. Okay, so the purines, this is kind of a joke, no offense against you Aggie-loving people, but what I always say is Aggies, A-G, are pure, not, just kidding, but I say Aggies are pure, A and G are pure, and then TCU, thymine, cytosine, uracil, I always think of colleges, TCU are pyrimidines. Okay, does that make sense? So if you want to remember a funny way to help you remember which ones are which, I know this is kind of corny, but I always remember, no offense against you Aggies, I always say it takes 2 Aggies, get it, the 2 rings, it takes 2 Aggies to change a light bulb, whereas it only takes 1 person from TCU to change a light bulb. It's kind of a diss against you Aggies, but I kind of do that joke to remember the double ring structure is an A or a G, my Aggies, okay, two people to change a light bulb, and my TCU, it takes 1 kid to change a light bulb. It's kind of a joke, but that's the way I do it. And then I also just kind of look at the longer word is with the single ring, pyrimidine or single ring, and then the shorter word, purines, those are the double ring structure, so it's almost like opposite. Okay, so here's your little breakdown. A single ring is TCU. A purine, which is a double ring, is an A and a G, because of that little Aggie joke. Just kidding. I like A&M. It's all right. And the last slide just kind of puts it all together, where you can see the last slide with DNA, and you can see in the middle of the DNA double helix, you know, because DNA is a double helix. So here's literally like one side right there. So this would be, ready? Here's your nucleotide, 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 nucleotide. So there's monomer, 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 all bonded together through phosphodiester linkages. But then this side we have the same thing, nucleotide, 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 nucleotide. But in the middle, we get those weak hydrogen bonds, which is a weak attraction between a positively charged hydrogen and something else that's electronegative. And that's because if you examine this, adenine and thymine, there are functional groups that are attracted to each other. So that gives us our double helix DNA. Make sense? Guess what? We are done. Great job.